Thank you so much for the kind introduction. Can you see my slides? Thank you so much for inviting me. I'm sorry I couldn't be there in person. I'm excited to talk to you today about methods for integration and hypothesis generation from high-dimensional biomedical microbiome datasets. Next-generation sequencing has fueled a deeper understanding of the complex community of microbes that live on and in the human body. My research has focused largely on the role of the gut microbiome in complex disease. Associations between gut microbes and many different diseases have been described, but a major challenge is to move beyond associations to causation and to determine the specific mechanisms by which microbes influence the host. A major focus of my work has been the role of the gut microbiome in the health of people living with HIV. The gut microbiome can affect many aspects of HIV disease pathogenesis. Since the gut is a major site of transmission of HIV because of transmission via anal intercourse among men who have sex with men, gut microbes are important for transmission because they affect the activation state of the target cell of HIV, CD4 T cells. Once transmission happens, other research groups have shown that there's a breakdown in intestinal barrier function, and microbial products and parts move from the gut to the blood, where they can drive systemic immune activation. With antiretroviral therapy, this is partially but not completely corrected, and remaining immune activation in people living with HIV has been linked with poor health and comorbidity. Thus, we are really interested to learn whether gut microbes can be targeted therapeutically to improve the health of people living with HIV. In my lab, we are also interested for a variety of diseases in developing workflows for discovering mechanistic links between microbes and health. Starting with in vivo observations in humans, where we develop multiohmic readouts, we seek to both apply and develop sophisticated analysis pipelines for integrating this data to develop hypotheses that we can go back into the lab and test, typically either in vitro assays using cultured bacteria and immune cells, or animal models such as notobiotic mice. These laboratory systems also provide us controlled environments where we also get multi-omic readouts, where we can get robust hypotheses that we can go back into our human data to test. Working with humans, however, is complicated. There are many, many different confounding factors that influence the microbiome, and these get compounded in disease when you have different disease severities and also treatments. As an example, back in 2013, when we published our first paper on the gut microbiome in HIV, we described very dramatic changes in the gut microbiome that we later learned were not driven by HIV. Specifically, when we added a cohort of men who have sex with men who were engaging in high-risk sexual activity for HIV, we found that many of the microbiome differences we had thought were driven by HIV were actually present in this population. As an example, so here's a principal coordinate analysis where we looked at the fecal microbiome of 217 individuals using 16S ribosomal RNA targeted sequencing. In this plot, the points are colored by green for men who have sex with men, orange for men who have sex with women, and pink for females. And the shape of the symbols represents whether they have HIV or not and whether they're treated or untreated. And as you can see, there's better clustering here by color than by shape, indicating a large effect of sexual behaviors. In this paper we published in 2018, we then did a better controlled analysis and established the differences in the microbiome that actually could be attributed to HIV infection, which were there, but more subtle. Within these systems, we really want to understand how gut bacteria influence key immune cell populations. So one key immune cell population we're interested in are T regulatory cells, because these are important cells for controlling pathologic inflammation, which is what we see in HIV and other diseases such as inflammatory bowel disease. So we already know that gut microbes can influence Treg populations by a couple mechanisms. One is through making molecules that they display on their capsule. So for instance, zwitterionic capsular polysaccharides are capable of inducing naive T cells to differentiate into T regulatory cells that can secrete anti-inflammatory cytokines such as IL-10. Other microbes can influence the same process by producing metabolites from the diet. So dietary fiber can be converted by gut microbes into the beneficial metabolite butyrate that also stimulates Treg differentiation. We have only scratched the surface in understanding the diversity of ways that gut microbes influence immune cell populations. So one goal is to develop a greater understanding of these processes through multiohmic data interrogations. So using, for instance, not only 16S, but shotgun metagenomics or microbial transcriptomics, we can look at bacterial composition and activity. By looking at the metabolome, we could determine molecules that microbes are producing. And then we want to integrate this or couple it with host immune phenotypes, such as what sort of genes are being expressed by host immune cells, or what sort of host immune cells are even there, using techniques such as flow cytometry or a more high throughput version of flow cytometry called CyTOF. We have been developing workflows for integrative analysis of these high-dimensional microbiome datasets. Here's an example workflow that a student of mine, Jennifer Fouquier, has been working on for longitudinal microbiome data analysis. But this workflow also has general properties in our non-longitudinal data analyses. So, for instance, on the left, we show the different sorts of data we want to input. So we have microbiome data, we have data from the host, such as complex immune cell population data, and demographic information. A first step we often want to do is to perform dimensionality reduction so we don't have so many features to work with. So the first thing I'm going to introduce to you is a method we developed for this in microbiome data called Scenic. Once you've performed pre-processing, there's also other types of pre-processing that you might want to do depending on your study design. So for instance, in this example, Jennifer was developing methods to calculate deltas in different ways between time points and longitudinal data. But at the end of the day, we often use similar methods to select features that are important for defining whether someone has disease or not, or that can predict disease phenotypes, such as random forest analysis or linear regression modeling. In my talk today, I'm first going to talk to you about a method called Scenic that we developed for dimensionality reduction in microbiome data. Next, I'm going to talk about workflows for identifying important features and relationships between features using combinations of random forest, linear regression, and network analyses. And then I'm going to talk about tools for post hoc analysis, so that once you have identified these important features, we can develop robust hypotheses that can be tested in the lab regarding why they may be important. So starting off with dimensionality reduction, I'm going to show you work on Scenic, which was conducted in my lab by Mike Schaffer, Kumar Thurimella, and John Sterrett. Scenic is a method for finding and summarizing highly co-correlated groups of microbes. Why would you want to do this? So one reason is it can increase statistical power. So rather than taking a major multi-correction comparison correction hit by correlating every single individual feature, we find modules of co-correlated features and sum them. This has the advantage of less statistical tests and also helps with downstream analyses that don't perform well with highly co-correlated feature inputs. Doing analyses in this way also can add biological insight because co-occurring groups of microbes can co-occur for interesting reasons. So there's two main reasons. They can co-occur for indirect reasons, such as having shared environmental preferences. And then also they can co-occur because they cooperate with each other. And that's particularly interesting if you have cooperative metabolism. In downstream experiments, it may be important to add this consortium or collection of microbes rather than a single one. Scenic is tailored to microbiome data sets. So there's other tools in the literature that have been developed, for instance, for gene expression analysis. But Scenic has a lot of microbiome specific considerations. So for one, when you're detecting correlations between all your pairs of features, the default method is Spark, which is a method that's developed for doing finding pairwise correlations in microbiome data that can accounts for sparsity. We have a lot of zeros, and compositionality. Everything sums to 1, and so you have relative and not absolute abundances. We sum our modules. There's many ways you can do this. We simply sum the counts of everything included in a module, and this preserves the total sequence counts, which can be important for downstream statistical analysis that also account for compositionality. A lot of these are sensitive to the total sequence count. And then it's also integrated into QIIME, which is a popular microbiome analysis tool, through a plugin. Correlation network generation is the first step. You just input a biome table or a table of counts of features across samples, and that creates correlation files that are then subject to module selection and summarization. So you get a new file that you can use in your statistical analyses with modules summed. And then it also produces GML files where you could interrogate the network topology using Cytoscape. This tool also has application to multiohmic analyses, so you can pick modules not only on microbes, but on metabolites. And so then when you correlate them, instead of asking whether an individual microbe correlates with an individual metabolite, understanding whether modules correlate with modules helps you do downstream analyses, such as whether microbes in a module alone or cooperatively can produce the metabolites in a module. We also gave a lot of thought to how we pick modules. There's a couple of different algorithms implemented within Scenic, but using real and simulated data that's described in a bioRxiv paper. It's in the second round of review right now, so hopefully a real paper soon. We became partial to an algorithm called the shared minimum distance algorithm, which is formed by taking this distance matrix of the correlation coefficients, making a hierarchical cluster, and then kind of drawing a line and forming clusters of things below that. This has the desirable property that everything within a module has a correlation coefficient threshold above what you define, and limits the negative property we saw in other algorithms of sort of a chaining where you wind up with O2s in the same module that are only weakly or even negatively correlated. This slide shows an application of Scenic to analysis of differences in the gut microbiome in men who have sex with men versus non-men who have sex with men. I noted already, we see really strong compositional differences here. And in this analysis, we ran the same comparison, both without first applying Scenic and after applying Scenic. In this heat map, we have everything that, we show everything that was statistically significant with ANCOM. The lower triangle is colored by the Spark correlation coefficient. So darker red is more highly correlated, and the upper triangle is whether they're phylogenetically related. And then on the kind of left and bottom panels where we have the names of the OTUs, we have a box if they were in the same module, a black box, and the module number. And then the orange box shows the features that were significant without applying Scenic. So I want to make two main points from the results of this study. One is applying Scenic did increase our statistical power in this particular case. So all of the OTUs that were picked by ANCOM as being significant without applying Scenic were recovered either as part of a module of co-occurring microbes or on their own. And then we also, though, you can see a lot of features there that aren't in an orange box. So those are all new features that came up that we did not observe before applying Scenic. But doing this also helps us with our downstream analysis interpretation. So as an example, module 2, which is kind of in the middle there, had a phylogenetically diverse group of different types of bacteria. And a couple of these were ones that we had actually already tested in the lab. So we had cultured some of these microbes because we had found them to differ in HIV-infected populations. And we used them to stimulate human immune cells that we extracted from blood. And we measured the ratio of a pro-inflammatory cytokine, TNF-alpha, to an anti-inflammatory cytokine, IL-10. A high ratio indicates a high pro-inflammatory potential of this microbe. And one of the microbes in module 2, Eubacterium biforme, which is now Holdemanella biformis, was particularly stimulatory of a pro-inflammatory signature. But Prevotella copri, which was a part of the same module as Eubacterium, did not. And so we took this to interpret that, perhaps Prevotella copri, even though we have found it before to correlate with inflammation, is not the driver, that Prevotella copri is co-occurring with other pro-inflammatory microbes that may be the true driver. So in summary, Scenic is a tool that allows for binning of co-correlated microbe features for downstream statistical analysis. It's built by microbiome people for microbiome people, so it plays nicely with tools like CHIME and considers things like compositionality and sparsity in the data. And application of Scenic allows for increased statistical power and biological insight. So next I'm going to talk about how we can develop broader workflows for identifying important features and relationships between them. And the first story I'm going to tell you about this was we published last year in M Systems, and it was analysis we conducted of the gut microbiome influence on metabolic disease in HIV positive and high risk populations. In this study, we recruited a cohort of 116 individuals. Some were HIV negative, some of the HIV negative were men who have sex with men, and some were men who have sex with women. And then we had HIV positive men who have sex with men who were either untreated or treated. We developed rich data from this cohort, including a lot of data on immune activation phenotypes in blood, the fecal microbiome with 16S targeted sequencing, diet with food frequency questionnaires, and on a subset of the samples, we had untargeted metabolomic data with LC-MS. We also had a lot of information on our cohort, such as their sexual behaviors, GI symptoms, and treatments that they were on. Our outcome of metabolic health came from metabolic parameters that we measured in a fasting blood sample, including glucose, insulin, and lipids. And these were used to produce a single measure of metabolic health. That it's a continuous variable that will be the primary outcome in all of the analyses that I'm going to show you. So in our analysis pipeline, we wanted to be able to use all of the complex data that we had, and also to understand that there were relationships between them. So for instance, on the left panel, showing that, for instance, the diet can affect the microbiome, which will influence the metabolome that can influence immune activation. So in order to have an analysis workflow that could understand individual predictive variables and also relationships, we did 3 steps. So in the first step, we used Scenic to identify and summarize highly co-correlated modules of features. And so we did this both on the microbiome data using Spark to identify the pairwise relationships, and on the diet data using Spearman correlations, because diet data also seem to form very nice modules of diet components that co-occur in different foods. We next used a flavor of random forest called VSERF for variable selection using random forest to identify features that predict the metabolic disease score. So VSERF is a nice tool because it not only gives you an estimate of how well it could predict the metabolic disease score, but also is optimized to report back the important features that the random forest used to make that prediction. Once we had all of these important features, however, one thing random forest doesn't do a good job of is kind of telling you how or why those features might be important. And so step 3 was to do post hoc analysis. And so in the network I'm going to show you on the next slide, all of the nodes in the network are different features that the random forest classifier identified as being important for predicting metabolic disease score. Each feature is colored by the feature type, so microbiome, diet, immune, or demographic. And the shape of the figure, if it's an up arrow, it was positively associated, so detrimental to metabolic health. If it's a down arrow, it's negatively associated, beneficial to metabolic health. If it's a circle, it was selected by the random forest classifier, but it was not linearly correlated with metabolic disease score. And then we have lines connecting the features. If they're blue, those features themselves are negatively correlated. And if they're red, those features themselves are positively correlated. And so don't be intimidated by this graph. I'll step you through it a bit. So I just want to show here that VSERF did select many different features from all the different categories in predicting the metabolic disease score. So the microbes are in blue, the immune are in yellow, the demographic are in purple, and diet is in green. And reassuringly, we did recover many things that had previously been described to be important for metabolic health. As an example, some BMI or body mass index was positively correlated with poor metabolic health. So since this is complicated to understand, we wanted to kind of delve into this network to kind of look at more specific hypothesis-driven features. And so I'm going to step back to a slide I showed you earlier where we was talking about our motivations. And so one main factor we're very interested in is bacterial translocation as a driver of systemic immune activation and comorbidity. And so the measure that we used for intestinal barrier dysfunction was LPS binding protein, or LBP. LBP is an immune factor that binds to bacterial products on immune cells in the blood. And so when we look at the nearest neighbors of LBP in this network, we get a very interesting picture. LBP was actually the single most important factor for predicting poor metabolic health, which was very in line with our hypothesis. And interestingly, LBP correlated with a lot of different things. It showed positive correlations with both BMI and age, and then also with a lot of other pro-inflammatory cytokines that we had measured in our study. But it also showed both positive and negative correlations with different microbes. And so our next step was to dig into the literature to try to understand or develop hypotheses about why this may be. And so two hypotheses we developed was one that LBP, we found it interesting that it negatively correlated with certain butyrate producers, such as those in the Coprococcus genus, because butyrate is not only anti-inflammatory, but it also has been shown in previous studies to support intestinal barrier function. We also found interesting a positive correlation between LBP and Doria. Doria, certain species can degrade sialic acid on mucins. Different mucin sialic acid profiles had been already described to be pro-inflammatory in HIV. And this identified microbes as a potential driver of this. And so we actually recently received a new grant to follow up on these hypotheses in the lab. So all these different links that I showed you in this prior slide were determined based on a simple Spearman correlation. But we also do a lot of work using linear regression in our human cohorts, especially because there's, like I mentioned, there's just so many different confounding factors. So for instance, on the bottom left, visualizing what you would do if you're using a linear cohort, to ask which microbes are associated with an immune subset while accounting for the fact that immune populations differ with HIV. And so we have 3 different lines. One is for HIV positive individuals in purple, pink is HIV negative men who have sex with men, and then green is HIV negative non-MSM. And we were able to detect a relationship between the bacterial genus Faecalibacterium and the immune cell population, CD8 T cells expressing a mucosal homing marker. But we were able to better detect them when we can account for the fact that that immune population was depleted with HIV. On the right, this is another type of model we often use where we introduce an interaction term into the linear model so that we can ask which microbes are differentially associated by cohort with which immune cell subsets. This could happen if, for instance, immune dysfunction driven by HIV leads the same immune cells to respond differently to the same stimuli. An example of this you might think of is you're hiking with your friend, they're going nuts because of the pollen, because they have allergies and you're not. That's the type of thing that we would try to detect using a regression model that allows for different response to the same stimuli with an interaction term. So a couple of years ago, we developed a tool called Volari together with Janet Siebert for visual analysis of linear regression results and applied them to looking at these interactions between microbes and immune cell populations. So the Volari interface takes outputs that from running linear regressions in the R statistical package and allows you to explore the data. So on the top, you have a network that you can interact with. You could click, for instance, on hubs that are immune cells attached to many different microbes. You can ask to show the network in the table below, or you can search the table and ask to show what's in the table in the network. The table shows all of the different significant associations found, gives data that came out of the linear regression provided by R, and also on the right, a convenient M-plot that you can click on. And when you do that, it shows you a detailed plot. And so this interface has been really super useful for us to look at carefully at the underlying data to ask, is this outlier driven? Should we have normalized the data in a different way? So I'm going to show you, briefly show you the results of applying Scenic to an analysis that we did where we had really complex immune data that we developed, that we generated from blood and biopsy data using a high throughput technique called CyTOF. And then we also had 16S microbiome data from the stool of the same individuals. And we performed pairwise linear regression models to look for associations between immune subsets in blood and biopsy, and then also between the microbiome and these immune subsets. So here's a prettier picture of the network that we developed. And what was really interesting is there were several immune populations that came up as hubs where they were connected to a bunch of different immune populations and microbes. So for instance, in a prior slide, I showed you how Faecalibacterium was correlating with CD8 positive, CD103 positive immune cells in the gut. So that immune population correlated with several different microbes, showing that they are influencing this immune population in the gut. And on the top, there's a really interesting hub. These immune cells are called mate cells, and these mate cells were correlating both with, the light green are blood immune phenotypes. So it's a blood immune cell, but it's correlating with a lot of gut immune cells, which are in turquoise, and gut microbes, which are in blue. And what was also interesting is when we looked at the detailed plots, we could see that this association between the mates and all of these immune cells and gut microbes were really driven or only observed in our HIV negative non-MSM populations. The mate cells were actually really depleted, entirely depleted in our MSM populations, whether they had HIV or not. So why were we so interested in this? Mate cells stands for human mucosal associated invariant T cells. They're enriched in tissues, and they have been shown to play a key role in mucosal barrier integrity, maintenance, and repair, which we're very interested in. They also are important for the control of HIV. So it's super interesting that these are depleted both in HIV positive and high risk individuals. And these mate cells have already been described to closely interface with the gut, even though they're largely observed in the blood, through specific microbial metabolites. So they respond to bacterially derived riboflavin synthesis intermediates. And our preliminary follow-up experiments have actually shown that this altered microbiome in high-risk MSM has less genetic capability to produce these intermediates that mates respond to. And so again, this is one that we have really been interested in and have begun to follow up in the lab for its potential implications in HIV control and transmission. So in summary for part 2, workflows that couple feature reduction with random forest, regression modeling, and network-based visualizations can allow for hypothesis generation and exploration of complex data. So ongoing work in my lab, we are streamlining these analyses using the SnakeMake workflow manager to add to robustness and reproducibility of our work. We're working on handling longitudinal data designs and applying it to a diet intervention we've done in HIV-infected patients. And we're also further developing tools for network visualization and exploration for networks from linear regression. So in the third part of my talk, I'm now going to talk about what do you do once you've identified these important features or these important relationships between features. How can you use knowledge in the literature to try to develop sophisticated hypotheses that are worth spending the time and expense it takes to test them in the lab? And 2 parts. First, I'm going to talk about a tool called Ammon that we developed, and then I'm going to talk about a new research direction I've been doing in collaboration with Larry Hunter and student Brooke Santangelo. And Brooke is here and giving a poster this afternoon. It's poster V024. So I'm going to just give you brief info, but if you want more, you should go talk to Brooke. So as I mentioned earlier, we know a lot about how gut bacteria stimulate certain immune cell populations. And so when we get a result of a correlation, it's really interesting to try to understand from what we know about these organisms in terms of their metabolism, what metabolites they can produce, or in terms of their biological activity that's been reported in the literature, whether these are what the mechanism of a link may be. And I already kind of showed you how I did this with this result. You know, we found microbes correlating with LBP, and we did a lot of PubMed searches to come up with hypotheses regarding butyrate and mucin degradation that we're going to test. So we wanted to develop tools that can better help with this. And one of the motivations for this was actually when I first took my first foray into trying to work with metabolome data in collaboration with Nicole Riesdorf. So we got a supplement to one of our grants to tack on metabolomics, and we worked with Nicole to generate untargeted LCMS data on a subset of individuals in that metabolic disease study. And one of the first things we did when we were trying to interpret the results was to look at all of these different small molecules and ask which ones correlated with the different immune measures we got out of blood. And so this is a heat map showing those results. The darker the red, these were positively correlated, and dark blue are negatively correlated. And these results for me were very difficult to interpret because I hadn't really ever heard of any of these metabolites except a couple. So for instance, indole caught my eye because I already knew that that was a microbial metabolite of tryptophan metabolism that had been shown in other contexts to be immune modulatory. And in our hands, it appeared to be pro-inflammatory. which is actually opposite of what some studies have shown. So, indole caught my eye, but a lot of these, when I Googled them, I was like, man, these don't have anything to do with microbes. And indeed, when you're looking at an untargeted metabolome, what you're seeing is a gamish of metabolites from a variety of different sources. And I should say this is actually the blood metabolome. So really, it could come from anywhere. It can come from microbes in the gut. It can come from host metabolism. It can come from elements of the diet or other elements of the environment, such as things you might inhale. And so how can we parse through these metabolomic data to try to focus on the metabolites that are truly influenced by the microbiome? And so one way we look to do that is by using information in KEGG, the Kyoto Encyclopedia of Genes and Genomes. KEGG has information on organisms, what genes they make, what proteins those genes encode, and the enzymatic reactions that those enzymes do. And so what we do is we input into Ammon a list of genes, either predicted or measured, to be in your microbiome, and then also the list of genes that are present in either the human or the mouse genome, depending on what system you're working with. And Ammon will determine whether the compounds that you've observed in your metabolome came from could have only been produced by bacteria. So for instance, compounds 14 and 15 on the right would be bacteria-only metabolites. Ones that could only be made by the human, so compounds 3 and 4 on the left, would be only made by the human, or ones that could be made either by the human or the microbiome. So those are both. So on the right is there's a Venn diagram showing the results of applying Ammon. So we have compounds on the right in green that were predicted to be only produced by bacteria. On the bottom in purple, predicted only to be produced by humans, and in the middle in blue, that could be from both. And then on the left, we have compounds actually annotated in our metabolomics data set. And so in pink, we have a bunch of compounds that we annotated. And you can see that there's a little bit of overlap here between what we observed in the metabolome and what we predicted to be produced by bacteria and host. But this overlap is very useful and interesting for helping us to hone in on compounds' origins. And so we do have, for instance, 40 compounds that we can say, that were observed that could have been made by bacteria only, 60 by host only, and 95 by either. So this is one of the methods that we applied when we were looking at which metabolites came from the microbiome in our study of metabolic disease in HIV. And so on the right, what we did was, we took metabolites present in 20+% of subjects, and that was 5,332. 416 of these could be assigned a KEGG ID. And of these 146, Ammon told us could be produced by our microbiome that we measured. Because we had such poor annotation, and there's clearly a huge gap in what we can annotate, we also took a notobiotic mouse approach. So we took germ-free mice, we colonized them with the microbiomes of our study participants, and we used paired t-tests to look at which metabolites were significantly different in a colonized versus a germ-free mouse. And in this way, we wound up with an additional 493 metabolites that were different and then also observed in our human metabolome. And there was some degree of sharing between what Ammon identified and what we identified in the mice, giving us a total of 603 metabolites of interest, because we had some evidence that the microbiome either directly produced them through Ammon or either directly or indirectly influenced them via the mice. So when we looked at metabolites that correlated with poor metabolic health, that metabolic disease score in our HIV cohort, we actually found a bunch, 150 metabolites correlated even after correcting for multiple comparisons. Of these, 68 could be annotated, and of those 68, 7 we determined to be microbiome associated. either because they were predicted with ammon, such as dehydroalanine, or because they were observed both to be predicted by ammon and to change in the mice, such as phosphatidylcholine and phosphatidylethanolamine. One of these I want to draw your attention to, though, is bacteriohopane tetrol. So this one was positively correlated, so predicted to be detrimental. It didn't come up with ammon or our mice, but the name itself, I looked at bacteriohopane and thought, I wonder if this has something to do with bacteria. And so searching the literature, I found that indeed it is an exclusive microbial product. And there was also evidence that this compound was involved in interesting pathways for metabolic disease. So why did our methods, ammon or the mice, not identify this? And so one reason, is that ammon actually did not uses KEGG, and KEGG did not have a gene attached to this metabolite to produce it. So, the last thing I want to bring up then is, that we still are very interested or dependent on looking at the literature. And so I've been working recently, collaborating with Larry Hunter and a new student in my lab, Brooke, to try to use knowledge to develop hypotheses. And so a prior student in Larry's lab had developed a database called Phenolator. Phenolator incorporates information on many different knowledge graphs and ontologies to describe paths between genes, proteins, metabolites, and disease. And what Brooke has been doing is trying to add information on gut microbes into this knowledge database. And so she started by adding from a publicly available database, Gut and Gene. It's a manually curated repository of relationships between gut microbes, microbial metabolites, and target genes from over 360 PubMed publications. And you can visit Brooke's poster to learn more about this, but by working this in, she could do shortest path searches. So for instance, on the right is an example where she was trying to explore a potential mechanism of an association between Faecalibacterium and Parkinson's disease. And the shortest path in her knowledge network suggested that this could be through the product benzoic acid and its influence on the pro-inflammatory cytokine IL-6. So in summary, microbiome composition has been associated with many different diseases, but an important goal is to develop a mechanistic understanding of causal relationships. High-dimensional and multiohmic datasets are important tools for hypothesis generation, and effective workflows must combine feature reduction, state-of-the-art methods for feature selection, and tools that facilitate results interpretation and hypothesis generation. And coupling these sophisticated data analysis methods with experimental validation has promise for mechanistic insights as to the role of microbes in disease. So I'd like to thank my lab. I kind of acknowledged people along the way who did the work, and then also my many wonderful collaborators, the study participants, and funding. So with that, I would love to take questions. Thank you.
Thank you, Catherine. It was great talk. Do people in the room have any questions? If you would do, please come to the microphone. Can you hear them? Can you hear us?
Can you hear me?
Yes, I can.
Hi, thank you for your talk. I was wondering, so when I received metabolomics data, I get a lot of unidentified metabolites. Do you have thoughts about how to integrate those into this kind of a workflow?
Yeah, and that is a huge problem. There are so few that get annotated, and one of our methods here was to just look at the mice, but obviously not everyone has that that capability to kind of colonize a germ-free mouse and define that. But the other tool I could think of is out of Peter Dorenstein's lab. If you have, for that you need MS data, but then they actually have ways you can set up kind of networks, and like it can help define, well, this wasn't annotated, but it's one methyl group away from this annotated thing. So there are ways to kind of, if you are really interested in one, dig a little bit more into what they are informatically, and then also things that correlate can be kind of targeted for further, definition of what they may be in the lab. But definitely, like, actually, when I talked to Stan Hazen once, he defined TMAO, and that's a really, famous microbiome story of a link between the microbes and metabolic disease. But he said when that first came up, it was unidentified. It's just so highly correlated that they just had to follow up and what is this thing? But it's a major challenge in the field.
Thank you.
Hi, thanks a lot for the talk. So when we analyze the relation between microbes and metabolites, we see a really strong batch effect. When we compare different experiments, we see really, really different relations. And very often when we compare to literature, actually we see in the literature different things, contradictory things in the literature, which again contradict what we see in different batches. So any idea on how to address that?
Yeah, and I mean, that is one thing that I critiqued about LCMS data, where, it really is hard to compare it between different studies. And I know in our studies, something like, it would have been great if we could have used existing data, that there's a lot of it where people have, compared like notobiotic to germ-free mice, but it is so hard to compare between studies and batches that we really were like, we ran it all at the same time, so that mouse, those peaks could be well matched. And so, yeah, that is a challenge with LCMS. I don't really have good solutions to suggest. I have, heard some people say that MS spectra are more easily kind of comparable. So we have been trying to move a little bit more towards that way. But then Nicole Riesdorf, my collaborator, tells me that method isn't as quantitative. So we kind of use a combination of those two.
Okay, thanks a lot.
One last question, and before you go, Catherine, there's a question in Q&A online. Can you answer that after the last first question, please?
Sure.
Thank you.
Yeah, this is a very nice talk. So my question is also simply related to the actually fact removal stuff. So in basically we have some metadata and we have some, we were trying to build the model. So when you try to integrate some metadata to remove actually fact, you basically in the model that you mentioned, do you basically just put the feature into the model, or you do first do some, for example, batch effect removal, and afterwards you put the basically purified features into the model. So which this process to prepare the which one are you using right now? I'm not sure whether my question is really clear.
I'm sorry that I had a little trouble understanding that.
Yeah. So basically, the question is do you just basically put all the features, for example, a diet or medication information into the model, or you just basically purify the other feature, microbial feature, using this metadata first, and afterwards you put this data into the model?
So I think you're asking kind of like when you're choosing which features to put into the model, are you kind of putting in everything or trying to purify or like limit them based on sort of prior knowledge of what might be important? Is that your question?
Yeah, something like that. Yeah.
Okay. Yeah. No, and that's an interesting. We've done a little of both because definitely when you're doing these fully exploratory analyses, you have the ability to potentially learn new things. But at the same time, you really need large sample sizes to make them work. And so, you don't always really have the power to do a truly exploratory analysis. And then, it really could help to try to narrow your focus or your interest in terms of only putting in features that you have some prior reason or hypothesis to believe might be important. So I actually really do vary those design decisions based on the size of our cohorts. If it's a pilot study, I just kind of want to, can I recapitulate what other people have seen or what is known? But if I do, have the ability to get like a big cohort, I get excited to be like, well, can we robustly find, some new thing we never thought of before by putting in, less targeting what we put into our analyses.